Hello, this is Jesse, and today I'm going to be going over RAST and gene finding in eukaryotes. And the objective for this tutorial is to use RAST to annotate genome of a prokaryote, which is Deinococcus radiodurans. And then next we're going to use anti-smash to mine for biosynthetic gene clusters and fungal genome. And then use GeneScan for predicting the locations and exon and intron structures of genes and genomic sequences. And we're going to be using a human sequence in this example. So first, we have RAST, and I go over getting a practice genome. So you go to NCBI, go to taxonomy, and then at NCBI, in the taxonomy, you can type in the taxonomy ID here, which is 1299. Enter, and then Deinococcus radiodurans, and you can click on the genome. Click in the box there, click go, and then you can select the genome, and then from there you can actually save the genome. In this example, I submit chromosome 1, so you can click on the chromosome 1 and you can save here, file, and then FASTA. Or you can do chromosome 2, and you can do chromosome 1, chromosome 2, and the two plasmids here. But in this example, I'm only going to focus on chromosome 1 just as an example. So after you save the file, you're going to need to upload it to RAST. So you can go to the RAST server here, and you should have already created your account already, and this time we're just going to be actually submitting a job to RAST. Once you open up the RAST link and you sign in to your account, you can go to upload a new job. Then you can choose a file and go through these steps that I have here for you. Just going to choose a file, and then you're going to just follow the steps to completing the submission. Use the taxonomy ID, Deinococcus, bacteria, species, radiodurans, and you can get all this information from the genome page of the organism. You can get the species, the genus, the taxonomy ID 1299, everything that's for, you can get it from there. Once you submit the job, you're going to have to wait for the annotation job to come back. Once it's done, we can view the organisms or the genome of the organism that's been annotated by RAST. And I already have this job done. So I can go to the job, go to jobs overview. And annotation might take a day or two, or it might take a few hours. It depends. So just keep that in mind when you're actually working on the homework. So here I click on annotation progress in this column, and I'm going to click on view details for the organism that I'm going over. And then we're going to go to browse annotated genome and seed viewer. Okay, and then from here you get some of the information about the size, the GC content, and the amount of subsystems. So subsystems are usually a group of proteins or coding regions that work together for some certain role. It could be the cell wall, it could be photosynthesis, RNA metabolism, DNA metabolism, and so on. So that's a subsystem. Usually there are going to be a couple or a few proteins in that subsystem. And if you scroll down here, it says subsystem feature counts. You can actually click on one of these plus signs to the left of each subsystem. So for instance, we can go to DNA metabolism, and we can go to DNA repair. This organism, Deinococcus radiodurans, is known for its ability to survive in high amounts of radiation. And one of the reasons why it's able to do that is because of its DNA repair system. So if you're interested in this DNA repair system, you can click on DNA repair, bacterial, and look at this particular subsystem or other subsystems. But I'm just going to show the first example here. And this is going to take you to a separate page. Once that loads. Okay, so once we get to the DNA repair, we can go to functional roles, and then you can actually look at the proteins that are involved here. And then you can go to subsystem spreadsheet, and you have the organism. You have a table here, and it gives you the protein that involved for this organism. It gives you other organisms too that have similar proteins. In this case, they might have similar proteins in this subsystem too. Okay, but we're only interested in the organism, our organism right here, so we can just filter it out. Just type in the actual name of the organism. Then we should have Deinococcus. Oh. Okay, so once we get the organism filtered out, we can actually click on one of the proteins that are in the subsystem. So we can click on Rec A, click on 2413, and then it's going to take you to the overview of this protein. So we can click on different things. We can view it in the Genome Browser. We can look at the sequence. So if you actually want to get the sequence in FASTA format, then here it is. You can download it. Or we can switch to the protein sequence. Here's the DNA sequence. We can go to Show FASTA. It'll give us the protein sequence here. Okay, and we can download it if you want, but we can also go back to the page. We can get out of here. We can view it in the Genome Browser. But first, let's actually go through here. So here is actually, green section is the protein that we're looking at. Right here, Rec A protein. And then it's showing you which ones are similar in this region compared to other organisms that have similar proteins. Oh, here's Rec A right here, the red. So right here. So this is showing you similar regions. Oh, we can go back. Here it is. Similar regions, Rec A, and where it occurs in other organisms. So that's if you want to look further into a particular protein sequence or a protein that's occurring in a subsystem for that organism. Additionally, we can go through here and look through the Genome Browser. So we're going to browse through the genome, the annotated features here. And you can filter based off of function, subsystem, but say we're interested in only hypothetical proteins, we can press enter. And here we have all hypothetical proteins, and look at the very bottom, it gives us the total amount of hypothetical proteins. Maybe we want to look at the protein sequence. So we can click on the ID here. We can click on sequence. You can also see here where this hypothetical protein has occurred in another organism. You can do a blast search, or you can run through Interpro scan. There's a lot of different tools you can use here. Go to the sequence, download the protein sequence here if you want to annotate it with Interpro. Copy, and then we go to Run Tool, Interpro Scan. And we can use Interpro to annotate it with any domains or motifs or any type of similar patterns that might occur in this protein sequence. Click Submit. Okay, so while that's running, we can also look through the KEGG metabolic analysis, which is what I have here in the tutorial. You can look at the metabolic network, and all you need to do is go to Comparative Tools, click on KEGG metabolic analysis, and you'll be taken to this page here, and you can actually look at the entire metabolic network of this organism. Lipid metabolism, carbohydrate metabolism, anything that was annotated for this chromosome. Okay, and you can additionally filter out for only certain metabolic networks. So just for like lysine biosynthesis, just like select. And then this will run. And you filter out only for the metabolic network of lysine biosynthesis. And in the tutorial, I look for this certain metabolic network here. You might be interested in something else like xenobiotic degradation or gluconeogenesis. So it just depends on what you're looking for, and you should be able to filter out based off of the subsystems or the metabolic pathways here, like phenylalanine metabolism, peptidoglycan, or lysine, whatever it is. So that's how you can do it from there. And then additionally, you can click on, from the very top where it says Job Details, you can click on View Metabolic Model. You click on View Metabolic Model, you'll be brought to another type of web tool here that you can use to filter throughout the data. Compounds, genes, compartments, biomass, and then pathways. You go through genes. It just depends on what you're looking for, but also gives you the reactions. And if you have this certain gene, what reactions it participates in, and then the function. So click on the reactions. You get the name. You get the compartment where it occurs. So it just depends on what you're looking for. In the homework, you'll be asked to look for certain metabolic pathways. You'll also be asked to look for hypothetical proteins and annotate them, right? In this case, Interpro wasn't able to annotate anything about this protein, so you might have to re-look. You might have to go back and look for a different protein or a different hypothetical protein. You'll also be asked about the total amount of GC content, so you can find the GC content here in the seed viewer. Right, you'll get asked the number of subsystems, or to look through a certain subsystem, right? You might be looking for stress response, maybe periplasmic stress, and then you can click on that certain stress response, periplasmic stress response, and it's going to give you the proteins that are associated with that subsystem, and you can get the protein themselves by going to their functional roles. So I'm going to get rid of this. Okay, so here's the lysine biosynthesis, but notice how you can filter out and only focus on a subsection of the overall metabolic network for that organism. Okay, but once this load, you will be able to have access to the subsystem here. And additionally, you can also download the file as a GenBank, the whole annotated file as a GenBank file, or you can download all the amino acid files, or all the proteins and amino acid FASTA format. You can also download it in GFF3 format. Usually you might be asked to download the proteins and FASTA file and then filter out those or do certain blast searches. So just know that you can always go through here, and whatever you're asked for, you should be able to download it from here. Okay. I've already shown over here, so we're just going to skip this part with Rast. Next, I'm going to be going over using anti-smash for a fungal genome. In this example, in this tutorial right here in the document that I have for y'all, I go over Candidiae albicans, but this case I'm going over a different one, Candidiae glabrata, and we're going to be looking at one of the chromosomes here, and I chose the actual genome this time. Click on the genome, and you will download the entire genome in FASTA format, but I already have it downloaded here. Here it is. So here's the entire genome, chromosome A. And then I also download it in GenBank format. So once you have it in either format, you can submit it to anti-smash. And this time it's going to be the fungal version. And once you submit it, it's going to be similar. This just how you would submit it for the bacterial sequence. Go to upload file, and you can click whatever you're interested in this tutorial, and in the document I just click these two, and then you would click this, but they're having an issue right up here, server move after outage, and they're not letting us use that feature anymore for looking for cluster border prediction based on transcription factor binding sites. But you could upload the file here, and I'm using this one. Upload it, and then you submit the job, and then it'll run the analysis. So in this case, I have the FASTA format here, and then I also submit the GenBank file. Okay, so the genomic file is just the DNA itself. The FASTA file is just the DNA sequences, and then we have the GenBank file, which is the annotated file from UniProt. Not from UniProt, NCBI database. When I submit the genomic, the FASTA file format, anti-smash will actually do the annotation for you, the gene finding. But if you submit the GenBank format, it will just go ahead and just look for the biosynthetic gene clusters and not have to do the extra step of doing the gene finding for you. But I already have the jobs run here. So here is with the GenBank file, I have 3 different annotated biosynthetic gene clusters, And then this next file, or this next run, I did it with the FASTA format. And notice how I only got 3 annotated biosynthetic gene clusters. Okay, in the first submit with the FASTA file that I submitted, it was actually able to pick, or the GenBank file, not this is the GenBank file I used, able to see that they predicted a non-ribosomal peptide synthase, or one that's similar or like one. And we can click on it. That's not what I want to go to. We can click on the file here. Here it is. Once we click on it, we can look at any predicted structures if there are any, and I believe for this one they aren't able to. But with this, we're actually able to get better results than what I have in this section. We're actually able to get, when I use Candida albicans, we get slightly better results with the non-ribosomal peptide synthase, and I also show using FASTA format and submitting a GenBank file. And notice that you do get different results. So if you do have the option to download the GenBank file, download that and then submit it to anti-smash instead of only the FASTA file format, because it could predict less compared to the GenBank file. So that's anti-smash. And then next we have GenScan. So we can open up GenScan. I have the URL there for you. So let's open up another browser, another tab, and then go over here. Submit GenScan. Once we get GenScan open, we can take the sequence. This case, we're going to be using a section from the human chromosome 8. And if I remember correctly, we're going to be using a section of the P22 and P21 right here. So we need to get the actual DNA ourselves. So we can just go back to NCBI, type in gene, and then I gave you a session number here that we can copy, paste, click here, and it's going to be this first one. It's going to be this one. Once we have this, we can go to FASTA file, and we can save the file. Okay, once we save the file, I already have it saved right here. Chromosome 1. Okay, we can submit it to GenScan. So let me bring it over here. Don't have it saved already. I thought I did. Okay, here it is. Human sequence. We're going to go to GenScan. We're going to submit the file. Open. Okay, then we can click on predicted coding domains and the peptides. Going to go to vertebrate, and we can say chromosome 8, P22, and P21, and run GenScan. And then we get our output. So here I provide a explanation of the output. So we have gene number and exon number here, gene number and exon number, and then we have the signal, so we have the poly A, then we have the signal exon gene, we have the promoter. From there, we have the beginning and the end of the poly A, signal, so this is the beginning of the end of that feature. We have the length, then we have the reading frame, and then from here we have the initiation signal. Then we have, from here we have the 5' splice site, and then the coding region right here, coding region score. This one right here, coding region score, and then we have the probability of the exon. And then we have the exon score. Depends on the length. And then the initiation signal, and then the splice site, and then the coding region score. But if we scroll down, we can also see the predicted protein sequence from the given coding domain, from that gene right here. But if we're only interested in the predicted peptides only, then you go to print options and just select for the predicted peptides only, and then click run GenScan. And then from there, we have all the, I think it's about 12 proteins that are being predicted from the gene sequence we got from the DNA section of the chromosome. We can actually copy and paste these into BLAST, NCBI BLAST. And we can run a quick blast search on them. And once we click run, it will do a blast search for all 12 of the protein sequences, and we can look through each one. In this case, I already know what protein I'm looking here for, but you might have to look through each one you might be interested in the proteins of a certain length. In this case, we're looking for any protein sequences, any CoreWe proteins that we did the blast search with that are less than 100 amino acids. And I will show you once this shows up. So here's all the blast searches that we did 12 blast searches. And we have a few right here. This is greater than 100 greater. We have, this one is less than 100, so we can look at that one. We also have this 56 amino acid one. And then some of them don't bring back any significant results from running just a quick blast search, but you could use BLAST-P. But I'm going to go ahead and click on this blast result right here with 94 amino acids. And then once the results come back, we can look at the top results and see what the identity of this protein is. And here we have, it looks like it's neutrophil defensin, which is correct. I did get the human chromosome section is already known to code for neutrophil defensin 1. And we can click on one of the top results. And you can see here, scroll all the way down, we can go to the actual UniProt page. And then from the database source, we can click on a session and we get to the UniProt page with the annotation information on here. And it is from neutrophil defensin, and it makes sense because that's exactly where I got this chromosome from, the chromosome sequence. So in these regions, there's a gene responsible for coding for the production of an antimicrobial peptide called neutrophil defensin 1. And that's it for this tutorial. Hope this is helpful.